Next, we have Dr. Greg Webster, who is a pediatric electrophysiologist from Lurie Children's Hospital in Chicago. Welcome, Dr. Webster.
Thank you for inviting me. Before I get going, just make sure the IT stuff is working. You can hear me okay, Marcia?
You're good, yes.
Okay, terrific. Well, thank you for inviting me. I'm honored to talk at SADS. I'm honored to be a part of the amazing work that has been done by the families who contribute. It's just a terrific opportunity. I've been invited to talk about gene therapy, and I was asked to talk about what gene therapy is and the state of gene therapy for SADS conditions. So I don't have specific conflicts of interest to report. So I think everybody who's on this call has heard this before, but I'll say it just to make sure that I start on the same page. This central paradigm that we think about is this idea that DNA, or the encoding part of the cell, creates RNA, which is the messenger between the DNA and protein. And the protein is the structural component of the cell. It's the stuff that makes a cell what it is. And then, of course, human beings are made out of cells. And I've put a little bit tongue-in-cheek a fish and an egg and a piece of meat here, but I think we all recognize that proteins on the interior of cells are really long strands of amino acids, or of the building blocks of proteins, and they're folded into a very particular shape so that the chemistry of the protein and the shape of the protein combine together to do one specific job or multiple specific jobs inside a cell. And that both the chemistry of that protein is really important and the particular shape of that protein is really important. Deleterious genetic variants that are not advantageous for cells or for people, cause disease. And in the left-hand panel, there's a DNA variant that I've marked in red, and that DNA gets translated into an RNA that is abnormal, and that creates an abnormal protein. And instead of being folded into the perfect shape to do exactly the job that it's been asked to do, either the chemistry or the folding of the protein is very abnormal or slightly abnormal. But one way or the other, it no longer does the exact perfect job that it was set out to do. And that results in abnormal cell function. And with abnormal cell function, the abnormal cell function eventually causes human disease. So this is the central paradigm that we're going to talk about as we launch into the idea of gene therapy. And I'm going to spend most of my time talking about relatively new gene therapies, but I want to bring it home to the idea that we've been using genetic medicine to make a difference in families now for a long time. So here's a family with ventricular fibrillation. This is a particular patient, and the top A labeled column is sinus rhythm, so the normal rhythm of the heart. And in the B column, or the B row, is ventricular fibrillation, a very disorganized, abnormal heart rhythm. And by the time we get down to C, or the bottom row of the three, the heart has stopped beating reliably, and now this person's in a lot of trouble. And this is the scary end result that we, of course, worry about in lots of SADS conditions, whether those are cardiomyopathy conditions, channelopathy conditions. This is the scary rhythm sequence that we are trying to prevent. And the family that I'm telling you briefly about is a pair of siblings that were 2 years apart. The first case, a 7-month-old who died suddenly, and the post-mortem, after the baby died, there were findings of cardiomyopathy. And in the second case, there was a newborn who had progressive ventricular failure and a ventricular fibrillation arrest, and despite really heroic attempts at medical support, didn't survive. And we did whole genome trios on actually both parents plus both kids, and actually got what we just heard so eloquently from Brittany, the idea of VUSs. And we looked into that a little bit further. And on the top panel, panel A is a normal human cell and a particular organ inside cells called mitochondria. And the proteins that make up mitochondria in this scanning electron microscope, this electron microscope image, which is very, very zoomed in on the cell, is you see the proteins lining up in rows as if you were plowing a field, and they're very well organized and they're all parallel to each other. In the bottom panel, which is from this family, those same organs, those same mitochondria have clumps of protein in them, where instead of being arranged in nice even rows, they're arranged in clumps where they can't do the job that they were set out to do. And this is the difference that I was illustrating between the nicely folded protein that does exactly what it's intended to do and the abnormal protein that clumps together. And in fact, when we look at mice and give them exactly that same variant, you find those same findings in the mitochondria. These are electron micrographs where the mitochondria are doing the same thing. So in this case, there's a new family outcome. We were able to screen the family's subsequent embryos for the C1QBP variants. This is the gene we found this abnormality in, and have an implantation of a fetus that had, or an embryo that had none of these variants. And now they have a 1-year-old who has a normal EKG, a normal echo, normal Holter monitors. This is something that many, many centers have been doing for a very long time. And although I'm not going to spend most of my time talking about this kind of gene therapy, I do want to point out that this has been making a difference in families for a while now, and we do have a place to go. That being said, this is my made up definition. People can quibble about individual sentences, but I think the type of therapy that people mean when they say gene therapy is to say the introduction of genetic materials into cells to alter the cell's function or behavior and ultimately influence human health. So that's where I'm going to focus. The idea of showing you the first slide is, first of all, to think about what happens with proteins, and then second of all, to think about what we're doing right now. But now we're going to move into more cutting edge science. So the disease mechanism, what happens in the cell, matters. And I'm going to talk about two different kinds of disease mechanisms, although there are more than these. And one is loss of function. So if you think about a protein as the working component of the cell, if you have a certain amount of workers, a certain amount of protein that is required to keep your cell functioning normally, a loss of function variant or a loss of function disease mechanism says, we just don't have enough workers. We don't have enough protein to do the work of the cell. That's what loss of function is. On the other hand, a dominant negative says, well, it's not just that we don't have enough workers, but the workers that are left behind are doing the work exactly as they're supposed to. The dominant negative says there's something about the protein that's left behind, or the abnormal protein that's left behind, that is deleterious to the other workers. Not only is it not doing its job, but it is getting in the way of the job that the other workers are supposed to do. And so it is knocking out or disrupting the work of the cell for the other normal proteins that would have otherwise been made. And I'm going to start by talking about this loss of function idea. And before I get into the heart, I'm going to point out that gene therapy is already pretty far along in a lot of cases. So here's an FDA-approved gene therapy. It's not the only one, but it's in the eye. And what happens is people lose vision because this RPE layer doesn't make a protein that is required in order to see. And a needle is injected directly into the eye, into the layer underneath the retina. And viruses that contain a code for the normal protein are layered on top of this cell layer. They go into the cells and they restore the protein that is missing, thus restoring vision. So when I talk to you about these next steps, I'm going to show you some of the pluses and minuses, but it's worth keeping in mind, we're not talking about science fiction. There are places where these therapies are not only conceptually possible, but are already being used and are FDA approved. So here's an example of loss of function, this kind of idea where there isn't enough protein in hypertrophic cardiomyopathy. And those of you who are very familiar with hypertrophic cardiomyopathy, which is a SADS disease, will know that loss of function is a big part of it. I'm simplifying this a little bit. There is some dominant negative effect in hypertrophic cardiomyopathy due to MYBPC3 as well. But I think the dominant idea here is this idea that there just isn't enough protein around. And so this is a study from 2014 in mice. And there were normal mice, and then they made a set of mice where they had changed them so that they had abnormal MYBPC3. And those mice with abnormal MYBPC3 made a heart that was affected by hypertrophic cardiomyopathy. So in other words, when they took away the normal MYBPC3, the mice developed hypertrophic cardiomyopathy. And then they added a gene back in to make normal MYBPC3. So first they took away the normal MYBPC3, and then they added it back in. And there were a whole bunch of pieces of this study that suggested that they were making a difference, but I'm going to show you the results in their study just from engineered heart cells, because I think they're the cleanest and the easiest to understand. In each of these three graphs that look at force and velocity, how hard is the heart contracting and how fast does it contract, I've put a green line under the normal mice. And the normal mice have a normal amount of contractility when you make a group of heart cells. When you give mice a phenotype that looks like hypertrophic cardiomyopathy, the force of their squeeze goes up, the velocity of their squeeze goes up. They develop, even in these engineered heart cells, a phenotype that looks like hypertrophic cardiomyopathy. And in fact, when they look at the mice themselves, there's some features of that as well. And then when they give that normal protein back, when they re-inject a gene that is then translated into normal protein, when they introduce that into the mice at day 1 of life, they keep the mice from developing that phenotype. And in this particular case where I'm showing you, they keep those engineered heart cells from developing that phenotype. So this is true gene therapy, right? This is the idea of identifying a gene that is not functioning correctly, where loss of function is the primary disease mechanism, and replacing it with a new gene that causes successful contraction in the heart. They did this by delivering a virus, and this is going to be a recurring theme, where they took a virus that is mostly emptied, and it's got little proteins on the surface, or actually glucose molecules on the surface, that determines where the virus goes, and in particular goes to a cell target in the heart. And if you cut open this inside of the virus, there's space to put other delivery cargo into. And in this case, what they're interested in is putting genetic delivery cargo into it. And the new DNA is the cargo carried by the virus. This is the central idea of viral vectors. Let's take a virus that goes to the heart, let's load it up with some kind of cargo, and then let's deliver it to the new heart, or to the heart. So by replacing missing protein with healthy protein, the idea is that you supply more workers and you cure this loss of function. But as we talked about, the cellular disease mechanism matters. So I told you about a way to do a loss of function gene therapy, and that was very successful in mice in this particular example. But what about in this case where the abnormal protein is actually damaging the prospects of the normal protein that's in the cell? So I'm going to focus on that next in another example of clinical gene therapy, or preclinical gene therapy rather, for inherited arrhythmias. So this is work by Dr. Ackerman's group. I know he's on the call with us. A normal cell handles potassium very well. And this is a diagram of a cell, and the circle in the middle of it is a nucleus. And it makes RNA, it makes DNA first, and it makes RNA that then in turn makes a channel that handles potassium, that causes potassium to move from the cell. But a loss of function variant decreases that potassium flow. So there's a variant in the DNA that gets translated into a variant in the RNA. And then not only is the protein abnormal when it's made, but it disrupts the functioning of the other normal proteins. This is that loss of function. This is that dominant negative effect that I was talking to you about. And although the data I'm going to show you is from an engineered cell line, the idea is that in the human heart, this is one of the mechanisms that causes long QT syndrome. And so the concept here is to actually do two things. First is to bind up the cell's own gene for potassium channel proteins to create a process of keeping the abnormal protein from being expressed. And in this case, it keeps any of the cell's internal protein genes from being expressed, or at least the one that most needs to be fixed, KCNH2. And then in that same, and that creates this loss of function idea. And in other words, the first step in this gene therapy is to induce the loss of function, to change from a dominant negative function to a loss of function type cell. And then in the same syringe, and in fact, in the same adenovirus vector, to replace it with the gene for normal potassium channel function. And then what you end up with is a cell that is now able to make normal potassium channels. And the authors of this study call this suppression replacement, or suprep therapy. And although I'm not going to go through this in detail, there are cardiomyocytes in this particular table that was abstracted from their work, where you have controls and you have 4 different lines of variants in KCNQ1. And here is a action potential duration. For the purposes of this, it is a marker that is similar to the QTC. And what happens is that by doing this suppression replacement therapy, you decrease the action potential duration, or you decrease this analog for the QTC. In other words, you are shortening the abnormal period that happens in these cells due to the abnormal mechanism of the gene. This is gene therapy for dominant negatives. It's the same viral vector concept. In this case, what they're delivering is both the ability to knock out the abnormal and normal genes in the cell, but also a way to supplement it with a replacement totally normal gene. I've kind of talked about in this talk 2 approaches, both a DNA and an RNA approach for replacement therapy, that was MYBPC3. I talked about allele suppression and gene replacement therapy for KCNQ1. There are a bunch of other kinds of gene therapy that are just started to get, that are, well, some of them have been worked on for a while, but that are just starting to come out. Modulation of signaling pathways, mutant gene inactivation, mutation repair, splicing, trans-splicing. This is a very complicated field, and cardiology isn't the only place that's happening. This is a disease approach that is being used in a whole lot of disease states. But cardiology is an area of active interest, and I think we're going to see a lot of forward motion in this over the next 5 to 10 years. I think it's really important, despite the fact that I've spent the 90th percentile of this time talking about how do you deliver gene therapy from a concept standpoint, to point out that we haven't solved delivery yet. There are genetic problems in delivery. How do you get these genes into the heart? How do you be sure that heart cells are actually the ones that are affected? In the particular the virus approach that I mentioned earlier, there's a problem with just how much you can get into that virus. There are other mechanisms involving engineered RNA that get around that, but there is a problem of how do you be sure that you can get bigger amounts of genetic data into the cell? And there's a big problem with pre-existing immunity that exists in humans that is less of a problem in mice. And in fact, way back in 1999, 30 years ago, when this all started, there was a really catastrophic case that set us back quite a while, where there was an immune response to the virus vector that was used. And unfortunately, it was a lethal immune response in a human. So while we've solved some of these problems and there are a lot of exciting approaches, I wouldn't say that we have solved the genetic problems. And then even when you get done with the genetic problems, there are just pharmacy problems, problems that we've solved in other drugs that are complicated by the fact that it's genetic delivery. What is the right dose of a genetic gene therapy? How do we make it durable over time? What about off-target effects, effects where that gene therapy doesn't affect heart cells, but affects everything else? In addition, it's relatively easy to think about how to deliver gene therapy to the inside of the eye. Although it seems daunting to put a needle into the eye, at least the cells on the inside of the eye are right there where you can get at them with a needle. The heart is a complex structure, much more complicated than the retina is. And it is filled with cells that need to be mostly delivered gene therapy. And in addition, that heart sits in the middle of the chest, and that chest is not nearly as accessible to us as the inside of the eye is. So on top of the genetic problems and the pharmacy problems, there's also some questions about the heart is a pretty protected organ, and it's difficult to get gene therapy delivered to it in a very reliable way. I don't want to pretend that there aren't really exciting processes of how to deliver that gene therapy to the heart. There are exciting mechanisms, but these are the state of affairs. So in summary, gene therapy is here for some diseases, and it's really exciting. Cardiac gene therapy for arrhythmia genes is here in petri dishes and still in animals. In other words, we can identify ways in which this is really successful, and it is not hard to look at the current literature for people who are trying approaches in heart and arrhythmia diseases and say, I see the future. And we can see the future with cardiac gene therapy for arrhythmia diseases, but we are going to have to solve the best approach for each type of genetic problem. get over delivery hurdles, pharmacologic hurdles, and then there are some regulatory and ethical hurdles that I haven't covered in this talk, which are likely to affect us in the long run as we start to think about how to approach this. In other words, it's complicated, but as with many things that are complicated, it's really exciting. And we may find that over time, we have the opportunity to really make a big difference in human health. And gene therapy is an area that I think most physicians are hoping will continue to blossom over the course of our lives. I want to thank the SADS Foundation for inviting me to give this talk. It is not my primary area of research, so I sit on a much more translational side than the folks that are doing this. So I want to be really complimentary to the colleagues who do the exciting research in this area directly in their labs. I've summarized this as if this is easy, but boy, this represents an enormous amount of work, an enormous amount of failed attempts along the way. And then I want to thank every family who contributes to research. I want to give a special note to the family with the C1QBP variants that were mentioned here and who allowed me to share their data with the larger community. So thank you for letting me be involved in the talk.
Thank you so much, Dr. Webster. And we have a few questions that have been posted in our Whova platform for Q&A. And if you can stay on a few more minutes, I'll just address those for you.
Sure, I'd love to.
All righty. I keep hearing 5 to 10 years for human trials. Every year I ask about the updates and I keep getting the same answer. What are the updates on gene therapy for having human trials?
I think that is a totally fair question. It's why I started with the gene therapy that's been FDA approved for the eye. There are gene therapies that are really being used, that are actually being used to cure human disease in people. I think you're right that we keep hoping that it will be within arm's reach. And it's why I kind of gave that slide summary at the end that there's a lot of hurdles. If you made me guess, my guess is that we will get it right in really, really, really narrow circumstances within 5 to 10 years. That would be my guess. My guess is that the very broad opportunity to affect a large number of families is probably longer than 10 years away.
Okay, another one here. Is gene therapy to be a one-time treatment, and can we take less or get off the medications we're on after having gene therapy, I guess?
That's a really sophisticated question, and I'm going to answer it in 3 different ways. First, as an example with the MYBPC3 that I gave, one of the things that does is it prevents disease from progressing, but it does not do anything to cure disease that is already present. And hypertrophic cardiomyopathy is different than long QT syndrome, is different than CPVT, is different than short QT syndrome. But I think there's still some uncertainty about in what circumstances are we going to be able to prevent progression versus in what circumstances are we going to be able to cure disease. So that's answer 1. Answer 2 is, in the perfect world, we would give you gene therapy, not you in particular, but we would give humans gene therapy, and it would take into every cell and it would last indefinitely and it would be very durable. That would be our goal. In the current formulation, there are some formulations, because the heart divides very, very slowly, because the heart divides slowly in adults especially, there are some formulations where we think we could deliver a therapy and it might be durable for a long period of time. There are other therapies that seem to be very short acting. In fact, some of them as short as hours. And that, of course, is not helpful when you think about a lifelong progression of disease. And when I talk about there being hurdles to delivery, one of the pharmacy hurdles is, how do you deliver a therapy that then is durable for the next 30 years? And we haven't totally solved that yet. And then the third answer to your question about what happens next in terms of getting off medications. I suspect that if somehow we were able to develop the perfect gene therapy that entirely fixed disease, that would be a goal. But one of the things that's been interesting about gene therapy is, in most cases, even in Petri dishes, even in mouse models, it substantially brings people back toward normal. But even in those models, it doesn't create exactly normal cells and exactly normal behavior. And I can't promise that gene therapy is going to be a magic wand that, even when it works the best, returns people to 100% normal. I think we're always going to be having the kind of risk benefit conversations and family discussion conversations where we say, here's what we know, and here's what we can predict, and here's what we think the risks are. And I think that's probably going to be a part of SADS type diseases for the foreseeable future.
Okay, thank you. Is this form of treatment something we may see in our lifetime, replacing the current regular forms of treatment of SADS conditions?
I hope so. I think if we get it right, we will get it right in some very specific circumstances first. One of the exciting things about the work that I showed you from Dr. Ackerman is the particular approach he chose, knocking away all the KCNQ1 in the cell and then replacing it. It does not depend on the exact particular mutation that you happen to have, meaning you don't have to make a gene therapy approach for every single mutation. And I think that's a promising approach, this idea of trying to think of ways to do it so we don't have to develop a drug for every single solitary mutation. But my guess is that when we get it to humans, the first time we do it, it will be in a pretty specific circumstance. And then it'll grow and we'll see more opportunities as we learn more.
Okay. If I have a gene therapy that fixes my variant, will my future kids be free of my SADS condition?
That's a really good question. I think when I hinted at that question of ethics at the end, that's one of the big questions that people have is most of the therapy or all of the therapies that I presented, the 2 therapies that I presented, they do not change your ability to pass genes onto your kids. They do nothing to alter your own production of sperm or eggs or what your chances are of passing that to the kids. When I showed that last slide where I talked about other gene therapies that are potentially possible, some of those could be used in ways that could potentially affect your kids or affect your kids' kids or affect generations into the future. I would say that most physicians, and I won't speak for them, but I will tell you that my personal thought is that is you need to be very careful about approaching that, because right now we think about that as a snipping out a genetic code and replacing it with another genetic code. But what we don't know fully is what those other effects of doing that are on other parts of the cell, on other parts of the genetic code. And therefore, how do we create a process of protecting what people call off-target effects that might affect your kids or your kids' kids? I hope that was a clear answer.
That makes sense. All right. Thank you, Dr. Webster. We really appreciate your time today on a Saturday and at the last minute here trying to pull it all together. We appreciate it.
Oh, I have been so honored to be part of SADS and have gotten way more from SADS than I could possibly give back. I appreciate everything that you guys have done.
Thank you.